Genetically engineering bacteria has the potential to lead to new solutions to problems such as pollution or delivering new types of treatments to patients. But releasing these man-made microorganisms into the environment is thought to be fraught with fears they may spread out of control. Researchers now claim they have developed a solution by turning bacteria into microscopic swarmbots. The bacteria have been engineered so they are unable to survive by themselves as they are reliant upon the protective proteins produced by their neighbors. However, if they spread out, causing their density to drop below a certain level, the bacteria can no longer get enough of this protein and quickly die. Dr. Lincoln, a biomedical engineer at Duke University in Durham, North Carolina who oversaw the study, said, "Safety has always been a concern when modifying bacteria for medical applications because of the danger of uncontrolled proliferation." Other labs have addressed this issue by making cells rely on unnatural amino acids for survival or by introducing a kill switch that is activated by some chemical. Ours is the first example that uses collective survival as a way of intrinsically realizing this safeguard. In research published in the journal Molecular Systems Biology, Dr. Yu and his team engineered a strain of the common gut bacteria Escherichia coli to produce a chemical called AL. The bacterial cells were also modified so that in high enough concentrations AL causes them to produce a protein that would protect them against an antibiotic. The researchers confined the bacteria within a permeable capsule and washed it with antibiotics. Provided the bacteria remained inside the capsule, their density remained high and they survived. But if the capsule was ruptured and the bacteria escaped, they spread out and were quickly killed. The concept works much like robot swarms where each member is reliant upon the others in the swarm for things like communication or positioning. Dr. Yu and his team said the experiment proves the concept of using the technology to create bacteria that are only able to survive while they remain in swarms. This would allow them to be released into the environment or given to a person without fear they will multiply out of control. Once the bacterial colony grows too big for its capsule, it will burst and release them, causing them to die. And in general, this concept does not depend on the use of antibiotics, said Yu. There are multiple directions we are hoping to follow with this platform. We're using non-pathogenic E. coli, but we hope to demonstrate that the same concept can be established with a probiotic strain of bacteria. Once we've established the platform, then we have the freedom to introduce whatever proteins we choose and allow these cells to engage in many different applications. Using probiotic bacteria could allow the researchers to respond to certain environmental conditions in a person's gut, to perhaps release drugs or nutrients when needed. It raises the prospect of using such bacteria to help tackle harmful food poisoning bacteria by programming the probiotic strains to react in their presence. Dr. Shuqiang Huang, another of the researchers involved in the study, said, "We can imagine programming probiotics that can respond to changes in their environmental conditions. That response could include delivering proteins or chemicals to modulate the microbiome. The technology could also be used to deliver bacteria contained within capsules that could help the body combat pathogenic bacteria," said Annalie, a student who was involved in the study. She said, "We want to program cells to respond to signals produced by pathogenic bacteria. We could inhibit their virulence and attack them at the same time."